Today paper for discussion is 9700 variant 1 to May June 2023. Question number 1 to 20 we will discuss, and next question number 1 is a graticule and a micrometer scale can be used to measure the size of biological structures that are viewed with a microscope. So here you can see that microscope is given. Number 1 shows the eyepiece. Number 2 is lens. Number 3 is stage. So according to this information, which shows the correct locations for the placement of graticule. Graticule is always placed in the eyepiece, and a micrometer scale on the microscope shown on the stage. Micrometer scale is always on the stage. So according to this information, B is the correct option. Now I would like to move towards the question number 2. And in question number 2, they have given us 6 different organelles which are present in eukaryotic cells. Number 1 is nucleus. Number 2 is mitochondria. Number 3 is centrioles. Number 4, rough endoplasmic reticulum, chloroplast, and at last it is Golgi apparatus. Okay. Number 1, a nucleus is involved in transcription. Mitochondria is involved in energy production. Centriole is involved in cell division. Rough endoplasmic reticulums have ribosomes on them, which is involved in the protein synthesis, and chloroplast is involved in the manufacture of carbohydrates, and Golgi apparatus is helpful in the uh, formation of glycoprotein and modification of them. So in this way, the organelle number 1, organelle number 2, organelle number 4, and organelle number 6, they are involved in the synthesis and secretion of a glycoprotein. So according to this information, the B is the correct option. Question number 3, and question number 3 is which cell structures can have messenger RNA inside them? Chloroplast have specific function in photosynthesis, mitochondria, nucleus. Endoplasmic reticulum don't have messenger RNA. They don't need messenger RNA. Only these chloroplast, mitochondria, and nucleus. As nucleus is involved in transcription, mitochondria in energy, and chloroplast in manufacturing. So in this way, these 3 organelles are involved in uh, in different functions, so they have messenger RNA inside them. So according to this, the B is the correct option. Now I would like to move towards the question number 4. And question number 4, you can see over here that there are a long description of the question is: a scientist carried out an experiment to separate the organelles in an animal cell by mass. Animal cell by mass. Scientists mixed the cells with a buffer solution which had the same water potential as the cell. The cells were broken open with a blender to release the organelles. The the extracted mixture was filtered and then spun in centrifuge at a high speed to separate the heaviest type of organelles. They sunk to the bottom, forming solid pellet 1. The liquid above pellet 1 was poured into clean centrifuge and spun in the centrifuge at a higher speed to separate the next heavier organelle. These organelles sunk to the bottom, forming solid pellet 2. This procedure was repeated for to get third and fourth pellets. What is the main function of the type of organelle extracted in pellet 2? Digestion of all lysosomes, for production of ATP mitochondria, production of messenger RNA nucleus, and synthesis of protein ribosomes. So, according to production of ATP, mitochondria is the second heaviest organelle in an animal cell, which produces a lot of energy and which stores the energy into it. So, option B is the correct option. I would like to move towards the question number 5. Which structures are found in the palisade mesophyll cells and prokaryotes, photosynthetic prokaryotes? Cell surface membrane, yes. Cellulose wall, no, cellular wall is not present because this is prokaryotes, photosynthetic prokaryote, which have cell wall made up of peptidoglycan. So cellulose cell wall is not present. Ribosomes and chloroplast. Okay, ribosomes are involved in the, and then chloroplast. In photosynthetic prokaryotes. So only number 1 and number 3 organelle ribosomes and cell surface membrane, they are present in it. So option C is the correct option. Next option, question number 6. Which polymers are present in all viruses and all prokaryotes? Polynucleotides, yes. Polypeptides, yes. Polysaccharides, no. So in this way, only polynucleotides and polypeptides are present in viruses and in all prokaryotes because polysaccharides are the carbohydrates. So here you can see that B is the correct answer. Only 1 and 2 types are present in viruses and all prokaryotes. Which set of steps, question number 7, which set of steps in the best method for conducting the emulsion test for lipids? Add emulsion test, add 2 centimeter cube of water in the sample, pour the water to a test tube containing, no. Add 2 centimeter ethanol in the sample and shake, no. Water to the sample and shake, no. Add 2 centimeter cubic centimeter of ethanol into a sample and shake. Pour the ethanol into test tube containing 2 centimeter water and shake again. Lipids are present, the mixture becomes cloudy. D is the correct option, in which the correct sequence of the test is given. All first 3 options are wrong options. Now I would like to move towards the question number 8. Yes, here you can see the question number 8 is the given. A student was provided with a solution of carbohydrate. They removed 2 samples from the solution and performed test on each sample as shown. Sample 1, Benedict test. Benedict solution remained blue. It means Benedict solution reducing sugar absent. Reducing sugar is absent. Second sample, boiled with dilute hydrochloric acid. Yes. Glycosidic bonds will be broken down over here. Neutralized with dilute sodium hydroxide. Benedict test. Benedict solution yellow, reducing sugar present. So it means it is which statement explains the results? Condensation reaction occur in sample 2 to release reducing sugar, not condensation reaction. There is hydrolysis. So glycosidic bonds in a polysaccharide have been broken to release reducing sugar. And sample 1 shows that sugar is present, no in not present. So B is the correct option. C, D and A are the wrong options. So B is correct option in this case. Now I would like to move towards the question number 9. And in question number 9, you can see which nucleus contain at least double bonds. Which nucleus contains at least double bond, and here there is sucrose, hemoglobin, no. Only collagen is that which contains the, so B is the correct option. Now I would like to move towards the question number 10. And what is question number 10? Which describes cellulose? Which describes cellulose? A branched chain of 1,4-alpha glucose. It don't have branched chain. A and B are wrong. And unreactive linear chain of 1,4-alpha glucose. 1,4-alpha glucose. Non it consists of 1,4-beta glucose. So D is the correct option in this case. Again, sorry. Now question number 11. Which part of the structure of hemoglobin carries oxygen? 4 polypeptide chains, heme groups, hydrogen bonds, hydrophilic R groups. In case of hemoglobin, it has to carry the oxygen by joining with heme group. So B is the correct option in this case. What is the maximum number of hydrogen bonds that can form between two single water molecule? Two single water molecule. Hydrogen, hydrogen, and the other hydrogen bond. So according to this, only one hydrogen bond will be present between two water molecules. So A is the correct option. Question number 13. Question number 13 is, CYP 3A4 is an important enzyme in an human digestive system, where it is needed to break down a range of different toxins. CYP34A has been shown to be reduced by substances called furanocoumarines and furane coumarines are found in some fruits, and so dangerous concentrations of toxins may develop in the human digestive system with fruits containing when fruits containing furanocoumarines are eaten. From the information provided, what can be concluded from molecules of the enzymes CYP3A4? Whenever these enzymes work, they lower the activation energy. So here in this case, in question number 13, the lower the activation energy of the toxin produced reaction, yes, they bind specifically through the active site to substrate found in the same food. They are the human digestive. They CYP3A4 is present in human digestive system, not in fruits. They change permanently when acted upon by a furan Q.1 molecules. They resume normal activity when concentrations decreases. So only here in this case, A is the correct option. I would like to move the next question. Question number 14. And what is question number 14? The fixed volume and concentration of a substrate and enzymes were mixed all over. All other variables were kept constant. The enzyme catalase reactions was left until it was complete, which graph shows how the rate of reaction changes with time. The rate of reaction and time, they are directly proportional to each other. When the time is increasing, rate of reaction is also increasing. Reaction changes with time. Rate of reaction with the time, it is increasing. Rate of reaction with the passage of time, but it becomes smooth after it. Rate of reaction, as the rate of reaction starts, the substrate concentration is decreasing with the passage of time because it is binding with the enzymes. So C can be the correct option, and in D it is increasing, rate of reaction is increasing, then it is decreasing. So C is the correct option in this case. A, B, and D are the wrong options. Question number 15. Question number 15 is, which molecule in cell membranes are typically involved in cell recognition? Cell membrane, cell recognition. Cholesterol is placidity for phospholipids. They are not involved in cell recognition. Glycoproteins and glycolipids and glycoproteins are involved in cell recognition. So according to this information, C is the correct option. And in question number 16, what can increase the fluidity of the cell surface membrane? Single bonds between carbon atoms in the fatty acid chains, cholesterol, longer chains, fatty acids. Only cholesterol is there, which can increase the fluidity of the cell surface membrane. So according to this, D is the correct option. Now question number 17. Question number 17 is, the three main factors that affect the rate of diffusion across a membrane can be expressed by the relationship shown. They have shown rate of diffusion is proportional to surface area and concentration difference. Rate of diffusion will increase when the surface area will increase. Concentration diffusion will also increase, but it will decrease when thickness of the membrane will increase. So they are directly, it is directly proportional to surface area and concentration difference, and inversely proportional to thickness of membrane. So which changes in the factors would result in the rate of diffusion doubling? Surface area, if the surface area has doubled, concentration difference has halved, there should be greater concentration difference. Thickness of the membranes are doubled? No. Thickness of membrane is halved? Yes. So 1 and 4 are the correct statements, and so C is the correct option in this case. Now, question number 18. Question number 18. Which student, a student measured the time taken for complete diffusion of a dye into agar blocks of different sizes which were suspended in the dye. The results are shown. 5 into 5 into 5 is 6.2, time is 6.2, and 10 into 10 into 10 is 16.1, 15 into 15, 34.5. So it means as the size is increasing, the time for diffusion is also increasing. And in the fourth case, they have asked what is the predicted time for complete diffusion of the dye into the agar blocks measuring 5 into 10 into 15 millimeter. So here you can say on once a 5, 10 and 15. So one side has is of 5 millimeter. So the time for diffusion will be the same as it was in 6.2 in first case. So option A is the correct option. Question number 19. Here you can see question number 19. Very long description of this question number 19. You can see there an experiment was carried out to investigate the effect of concentration of sucrose solution on cells in a plant tissue. A sample of plant tissues was cut into 7 cylinders of equal length and diameter. The mass of each cylinder was recorded. Each of the 7 cylinders was put into different sucrose solution concentration. After the 2 hours, the cylinders were removed and blotted dry and reweighed. The percentage change in mass of each cylinder was recorded, and they have shown us a graph. The graph shows the results of this investigation, and we have to find which can row explains the results if plant tissues cells were put in a sucrose solution of 0.45 molar decimal. 0.45 will be here. So it will be here, and we will draw in this way. And then water potential of the cytoplasm of the cells at the start of the experiment compared with the water potential of 0.45 sucrose solution. So it will be less negative. So any one of these. And change in the volume of the vacuoles of the cells at the end of the experiment that were initially placed in sucrose solution will decrease. So it will decrease. So it will decrease. So answer is A. A is the correct option in question number 19. Now the question number 20 is there. You can see in question number 20, the diagram shows part of the organization of a section of DNA molecules and the associated histones P and R. Histone in phase of mitosis. So which statement about the feature labeled P, Q, and R are during the prophase of mitosis is correct? So here you can see which statement about the coiled DNA molecule forms Q and wraps around histone R to form small clusters held in place by the histone P. No. P and R are wrapping. The groups of histone P and its associated DNA Q. No. Moves closer together as the chromosome condenses around R. No. The histones P and R are made of protein around which the DNA molecule Q is wrapped so that the DNA molecule can fit inside the nucleus. The linked groups of histone P and R and the associated DNA form strands but that fold and twist together to form a chromatid. So this is the correct description of the picture given up. So D is the correct option in this case. It's students video is completed. We have discussed 20 MCQs. In the next video we will do the question from question number 21 to 40. Please like, share and subscribe my video. Thank you so much.